science class. Here is a very, very exciting YouTube on a scientific method. And the scientific method is essentially what you read about, we'll be doing in class, is just steps involved in trying to solve some problem, observe some phenomena, try to explain what's going on in life and in nature. And basically it's the fundamental aspect of science, trying to explaining things. And of course you need to start out with making observations. Generally speaking, the next step is making a hypothesis. You test that hypothesis by doing an experiment. You collect data during the experiment and you come up with some conclusion. And hopefully when you're done you'll go, was I right? Let's check that again. And then if you can retest it and come up with the same conclusion, or better yet, other people can come up with the same conclusion, then you might be on the right track. So let's go through these really quickly in a little bit more detail in terms of what they all mean. Okay, so observation, pretty straightforward, we talked about that already, whereby you use your senses, taste, smell, sight, etc., and try to collect information. Now oftentimes, as in this little figure here, you may use an instrument like a microscope or telescope or rulers to get a little more detail, but it's still just collecting information. And so for example, you might look at a pond and see some salamanders nearby, and you notice that instead of having straight tails, their tails are curved a little bit. Next step is hypothesis. So based on your observations or this problem or whatever issue you're trying to figure out, you come up with some type of explanation. Now you always hear about an educated guess, which is fine, but it's some suggested problem, some solution to the problem. But the important thing about it is hopefully it'll set up some type of way to test that. So in other words, a hypothesis oftentimes may be testable. They aren't always written this way, but sometimes you can use this if and then. So if I do this, then this will happen. It kind of forms the basis of a hypothesis. If I see this pattern occurring, like in the box you're doing in class, and I think purple will be in the bottom because we have all the primary colors but only part of the secondary colors, that type of thing. And again, the last part kind of goes with that is the fact that it kind of predicts something, which then you can maybe test. So let's go back to our salamanders. So here's a possible hypothesis that I think the reason those salamanders have curved tails is because there might be some type of contamination or pollutant in the soil where you find them living. Next step is experiment. And of course, the whole point is to test the hypothesis. Now we've talked about variables already. Remember, variables are those things that can change in experiment. Some are important, some not so important. So that's, and we talked about the experimental variable. That's the one actually testing to try to, like, let's say I want to see what effect does light have on plant growth. Well, the experimental variable in this case would be light. And as we already know, that if you have too many variables being changed at once, it's not much of an experiment. So you want to have what's called a controlled experiment. We only look at one variable at a time, and oftentimes you compare it with something. So controls and variables. So an experimenter changes one factor, observes or measures what happens. That's basically what I just got done saying, but here it is again in another little slide here. And that is actually me. Notice the hair. Okay, the control variable. The control variable is the one you can make comparisons to. It's kind of your point of reference. So to make sure that you don't change that and that you compare it to something else. And these factors, hence here, called control variables. And I just got done mentioning that the whole purpose that you have a control is that you want to, like, it's basically a point of comparison. So let's go back to, like, let's see what effect green light versus red light has on plant growth. Well, you'll need some type of point of comparison of just plants growing in regular light without those variables being involved. So point of comparison. Now we have already talked about this in graphing, that we have independent and dependent variables. You might recall that the independent variable is the one that the researcher can manipulate. You kind of know that going into your procedure. And the dependent variable is the one that you collect oftentimes or observe. Then we already remember that independence on the x and dependence on the y. But it's more important to know the difference between them in words in terms of what they mean versus what axis they go on. So, for example, suppose you want to figure out the fastest route to walk home from school. So what's the quickest way to get from your house to school? Not that you'd want to get there fast, though you want to get to school fast, but take your time getting home, no doubt. So you try different routes on the way and time how long it takes to get there. And since you're only interested in finding the route that is fastest, you'll do the walking yourself. So in this situation, the independent variable would be what you're changing that you have control over, that'd be the different ways you go. So I go up 611, or I go some back way, or whatever it happens to be. You're changing that, you're manipulating that. And the time it takes to get there is kind of essentially what you're collecting, a dependent variable. And you want to make sure that all the variables are the same. So in other words, you're the one that's doing the walking this time. If you had someone who was a power walker, that might throw things off a little bit. So you want to make sure that all the variables are the same except for one, in this case the route that you're traveling. And this should go without saying that when you do experiments, you want to do them over again several times. Like if you want to figure out what effect does this fertilizer have on plant growth, you just don't grow one plant, you grow several plants, and you may repeat it several times. Lastly here, so 2 groups are required to control an experimental group. And this gets back to the idea that you only want to test one thing at a time, one variable at a time, kind of like the seaweed example I gave you with that homework. Now, the output of most any experiment is going to be the data that you collect. And you might recall that there's essentially 2 different types of data, both equally important. One is quantitative, where you use numbers, and more than that, you might want to use units too. In other words, if I walk outside and say it's really hot outside, well, that's not quantitative. If I say it's really hot outside because it's 95 degrees Fahrenheit, that would be numbers, and that'd be units associated, versus just qualitative, it's kind of hot out, or it's muggy. It could actually give you the relative humidity and percentages and things. When you collect your data, and this is a problem that I see a lot, is that it's on a scrap of piece of paper or just kind of written down somewhere. Organizing them in tables are very helpful. Charts and graphs kind of put it together, also another layer, but just collecting data neatly is important. Then the last part of this would be, based on your data you collect and the hypothesis that you're testing, you come up with some type of conclusion. And this is oftentimes the hardest part, to make sense of your data. And last but not least, as I said before, that generally speaking in science, we want to verify the results by having other researchers test them and retest them. In other words, is your information reproducible? Is it valid? So run down through these real quick again. It's identify the problem, and that's oftentimes based on observations. Form a hypothesis, design an experiment to test the hypothesis, collect data, form a conclusion, and last but not least, retest. So that would be it.